Sponsoring this new fertility series is Let's Get Checked. They offer home testing kits for sexual health, men's health, and women's health. A kit gets sent discreetly to your place. You collect a sample and send it back. It was really easy to get the results. I got an email notifying me it was ready, and I popped on the website to read my results. I did their female fertility test to help me understand my hormonal health. For 20% off your own kits, use code DNA Today at checkout on their website, letsgetchecked.com. Again, that's letsgetchecked.com with code DNA Today for 20% off your kits.
How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hello, you're listening to DNA Today, a genetics podcast and radio show. I'm your host, Kira Deneen. On this show, we explore genetics' impact on our health through conversations with leaders in genetics. These are experts like genetic counselors, researchers, doctors, patient advocates, and more. We are nearing the end of our infertility series here on DNA Today. If you haven't already, listen back to the last 5 episodes to hear from a fertility genetic counselor, a couple who went through reciprocal IVF, parts 1 and 2, filmmakers from a fertility film called ANYA, and last episode, a medical director who shared her experience on diagnostic testing for fertility. My guest on this infertility installment is Jennifer Eccles, who is the head of genetic counseling at Genomic Prediction Clinical Laboratory. Jennifer has over 20 years of experience in reproductive clinical genetics at institutions like Columbia, Beth Israel Medical Center, and BioReference Laboratories. As a licensed and certified genetic counselor, Jennifer has spent her career discussing genetic testing with individuals and couples at various stages along the path to parenthood. In her current role, Jennifer provides genetic counseling services along with authoring clinical and marketing content. Jennifer's publication history is focused on the patient-facing aspect of genetics and clinical testing. She's also a fellow graduate of Sarah Lawrence's Human Genetics program. Thank you so much, Jennifer, for coming on the show. I'm really excited to dive into PGT-P. But before we do that, could you give our listeners a quick refresher on what are the types of PGT to give them more of an understanding of the context that we're talking about?
Yeah, definitely. And thank you so much for having me too. This is super fun and exciting. So PGT stands for pre-implantation genetic testing. It used to be, and is still sometimes referred to as PGD for diagnostics or PGS for screening, pre-implantation genetic screening. But now it's often referred to as PGT. And then usually there's a dash and other letters following it. So PGT-A stands for pre-implantation genetic testing for aneuploidy. And that's the most common use right now of PGT, and that's screening for aneuploidy, which is looking at the number of chromosomes in an embryo biopsy. We have PGT-SR, which stands for structural rearrangements, which is geared for couples where one member of the couple has a chromosome rearrangement, and that couple is at risk, higher risk than the general chance for creating embryos with extra or missing chromosome material. And so that's what that test is designed to address, that issue. And then there's pre-implantation genetic testing for monogenic diseases, PGT-M, and that's designed for couples who are concerned about and at risk for different single gene disorders, genetic changes that impact one particular gene. And those conditions include things like cystic fibrosis, Tay-Sachs disease, sickle cell disease. There's thousands and thousands and thousands of different genes that do different things in our bodies. And so PGT-M addresses those types of conditions. And then PGT-P refers to pre-implantation genetic testing for polygenic disorders, which is a whole different ball of wax. Go ahead.
And I was going to say, before we connected, I had never heard of this. I've done this fertility series now for a couple months and exploring all these different areas of genetics and fertility. And then we connected and I was like, what is this? I've never even heard of this being used. So tell us, what is this? First, what polygenic conditions are and a little preview of what we're talking about.
Yes. So And it is new in terms of the IVF world. This is brand new testing. In terms of polygenic testing in general, this has been around in used in other ways. But polygenic testings in general are conditions that are influenced by, instead of one gene, instead of a monogenic condition, multiple genes. And so when we think of polygenic conditions, we're thinking about things like type 1 diabetes or risk of schizophrenia or certain cardiac disease risks. Cancers can be polygenic. So those types of conditions. And so the science behind polygenic testing has been around for forever and a day, but its use in embryo testing is brand new. And that's what's exciting about, working in the field that I do and getting to learn how this can be used in this way, in this brand new way.
And so when you say it's brand new, how new are we talking? It's brand new 2020, brand new this month?
Brand new 2019.
2019, wow.
Yes, in terms of the science, from a PGT perspective, a little bit longer than that, but the actual first clinical cases where we issued clinical reports for PGT-P studies was in 2019.
And so you mentioned a couple of the different polygenic conditions just to get a sense of what a polygenic condition is. And as you said, it's something that doesn't just have one genetic cause, like cystic fibrosis, we're looking at a certain gene and saying, okay, is this gene have an issue? And then you would have the clinical diagnosis of having cystic fibrosis from there. But for this, you're looking at so many genes all playing a role into this condition, along with other factors like environmental factors that you mentioned. So obviously there's so many polygenic conditions. What polygenic conditions can actually be screened using PGT-P?
Yeah, good question. So right now, clinically, the conditions that are available for screening include type 1 and type 2 diabetes, schizophrenia, certain cancers like breast cancer, testicular cancer, prostate cancer, malignant melanoma and basal cell carcinoma, so certain types of skin cancers, and then coronary artery disease and heart attack risk, hypertension and hypercholesterolemia, which are also related to those risks, and then type 1 diabetes and type 2 diabetes. So there's a bunch of different conditions. You're right, there are so many different polygenic conditions, but screening right now is focusing on that particular group of conditions for a couple reasons. One, because those conditions have significant impact on general health, which is what the goal of polygenic testing is designed to do. It's designed to help give information so that couples can choose and decide which embryo to transfer by using this information. And the other reason why those specific conditions are included is because those conditions are so common that we have a lot of data where we're able to generate enough data to be able to create polygenic risk scores and use that information for our testing. So the reason those conditions are in there are sort of twofold, because we have a lot of information to be able to design the test for them, and because we think that there's value in having information about those specific conditions.
And when it comes to this test, one thing that often as genetic counselors, we're explaining the difference between screening and diagnostic testing. And so this has become second nature for us. But for those that may not understand, why is this screening and not testing?
Yes. And that you're right. That is a very, very important distinction. And it's important that any couple coming for this type of testing knows what that distinction means. So diagnostic testing is what we would say more for screening for monogenic diseases. It's being able to give as close to possible a yes or no answer. So for monogenic testing, we say the testing is greater than 99% accurate for being able to say, yes, this genetic variant is there, or no, this genetic variant is not there. And we know if the variant is present, that we're concerned about that disease, and not present, then we're not concerned. And it's not quite black and white, but it's as close to black and white as we could get in PGT testing. And so diagnostic testing is really, that idea of getting as close to a yes or no answer as possible. Whereas screening is being able to more address risk. And this testing, particularly polygenic testing, it is not diagnostic testing at all. The way that the testing is designed is for us to be able to say whether risks for particular conditions are higher or lower than the general population. And based on that combination of risks, then couples can use that information to be able to say, well, based on the risk profile in this particular embryo, we're going to prioritize this embryo for transfer and maybe deprioritize this embryo for transfer. And so it's not a test that can guarantee the presence or the absence of disease at all, but it can be used to help couples reduce the chance of those conditions developing over an individual's lifetime and their child's lifetime.
And it's taken time to be able to accumulate enough data to be able to do this testing, as you kind of alluded to before. How does the science work behind screening for these polygenic conditions? I mean, you're not looking, as we said, at one gene. How many genes are we talking about for this?
Right. So when we're thinking of polygenic testing, we think more in terms of single nucleotide polymorphisms. So there's not a particular number of genes that are included in this platform, but rather hundreds and thousands of SNPs that are tiled all across the genome, like 800,000 different SNPs that are located in tiled across all of the genes along the genome. So the way that the test was essentially designed, because you do need a lot of data, you need data from hundreds and thousands of people. So the first thing that the developers of the test did was they went to the UK Biobank, where there's a million different samples of individuals, and basically they looked in adults and they said, okay, well, of the adults who have breast cancer, type 1 diabetes, cardiac disease, whatever, of that, can we look and see what the similarities and dissimilarities are in the profile, in the SNP profile of people with these conditions? And using that data, was able to extrapolate that data and design and create all of the bioinformatics and all of the magical stuff that is definitely, as a genetic counselor, really truly magical to me, in the sense that from a math and calculations perspective, is fascinating to listen to them talk about how they come up with the calculations for these things, but essentially come up with the calculations to be able to figure out what these polygenic risk scores are, which is a way of using this genotype information to determine specific risks for these conditions. And what we're actually working on now, that was sort of the first iteration of the test, and that is still the bones of the way that we're doing the test right now. But in addition to sort of figuring out, okay, well, what are the risks of specific individual conditions like type 1 diabetes or what have you, but also looking and figuring out, well, can we sort of put sort of a general risk, or maybe make this number more holistic? And so we're starting to look at these numbers, not just necessarily in an individual way, because maybe it's important to look at individual diseases in some families. Maybe, for example, a family may be especially concerned about type 1 diabetes. They may be very interested in that particular number. Or another family may say, oh, I'm very interested in looking at risks of schizophrenia because we have schizophrenia in our family. But other families may not be so interested in looking at very specific numbers for, let's say, hypertension or hypercholesterolemia or other numbers. Maybe for that family, it may be better to have more of a single number that takes all of these sort of risks and puts them together to sort of help identify more of a global risk estimate. And that's what we're looking at right now and what we're going to be, clinically starting to do very, very shortly. So it's a way of taking all of these numbers and putting these numbers together to be able to say, okay, if we have 5 embryos and of these 5 embryos, which can we say, and assuming all of them all of these embryos are euploid, because that's an important piece of the puzzle, aneuploidy screening and looking at the number of chromosomes always occurs in all of the testing that we do when we're doing polygenic testing. We're looking at, let's say we have a group of 5 embryos and everything else is the same and they all look euploid, then can we use this additional information, this additional polygenic risk score information to help a couple decide based on this information of this group, which has the lowest relative risk of these diseases as a group, and that embryo could be prioritized, and then which is the next and which is the next, so to sort of help decide which embryo to choose. Because right now we have, if we do aneuploidy screening and look and say, okay, well, we have all this group of embryos and these embryos are all have the usual number of chromosomes, what's sort of the next way? What else are we going to look at then? Right now, we look at morphology. We look at maybe a couple of other pieces of information. Some couples may make the choice based on embryo sex. But this is another piece we're thinking of as another useful way, a useful piece of information to be able to help couples triage their samples. And so, but that was getting sort of to the original question. Yeah, the genesis was the UK Biobank. And we're also going to be looking at, other biobanks and gathering as much data as possible as time goes on. And as polygenic risk scoring becomes more of the norm in general, that data will help, because that will also help us be able to populate that data into our testing platform.
The more data you have, the more accurate these risk assessments become, and it gets more tailored to that. It's so impressive and really mind-blowing when you think about it. You're looking at an embryo, and you're able to do this polygenic risk score for all of these different conditions. As you said, you're saying, all right, let's look at the genome, but not the whole genome. We're looking at all these specific pinpoint spots that accumulate to then give someone a higher risk for one of the many conditions you mentioned. How is this even possible? Like, what is the process? You're going through IVF to be able to do this, but how does this work in terms of, for the patients to understand, what are the steps for them to be going through to be able to have this be done and have these analysis ready for them to then be able to choose the best embryo to first try to implant?
Yes. So you're exactly right. So in order for this testing to occur, it has to be part of an IVF cycle. And so a couple undergoing IVF for whatever reason, either because they're going through IVF to treat fertility issues that they're having and they're interested in testing for aneuploidy and potentially adding polygenic testing, or for a couple who doesn't have necessarily fertility issues but is interested in addressing genetic risks in their embryos for whatever reason, would go to a reproductive endocrinology group and initiate IVF. And so in order for this to happen, couples would undergo essentially a retrieval cycle, which is taking all the medications and doing everything necessary in order to retrieve eggs. And then the sperm and egg come together. And then the cells of that embryo, that developing embryo, grow and divide for about 5 or 6 days. And at that time, they do something called an embryo biopsy. And they remove about 5 to 10 cells from the embryo. And those 5 or 10 cells go to the genetic testing laboratory, so a place like Genomic Prediction. And then we run the testing. And so it takes typically about 2 weeks to issue a report. And then the report is issued to the couple's IVF doctor. And then those couples, along with their doctor, can review for each embryo all of that data, and then use that data in decision making to decide which embryo to transfer. And then the couple has a transfer cycle which is that whole cycle designated to taking that embryo and to transferring that for hopeful implantation. And so it does involve quite a bit in terms of, it's very heavily a piece of that whole IVF process.
And you mentioned that for patients or couples that are maybe have not experienced infertility, this is an option that if they wanted to look into this. But for people that have experienced infertility, could this possibly help with their infertility, or is it really a separate option and process?
I wouldn't say it's separate in the sense that, for many couples going through fertility, aneuploidy screening is often an important piece of the puzzle for that couple, because aneuploidy is one of the most common causes of implantation failure and miscarriage. And so if a couple is already pursuing PGT-A, then having PGT-P doesn't actually really change anything from a testing perspective, in terms of needing an embryo biopsy and waiting for PGT report to come out and those types of things. The only additional samples we would receive are saliva samples from the patient and partner. So in terms of being a separate process, not necessarily. In terms of the question of does this have the potential to help couples who have fertility issues, in terms of fertility, we're actually very interested and curious, and intend on studying this. Right now, we don't know if polygenic risk scoring, in embryos transferred with better relative polygenic risk scores versus poorer polygenic risk scores, do any differently in terms of implantation. And it's actually something that we plan on studying as part of an IRB approved study in the near future, starting at the end of this year. So we don't know the answer to that question yet. Right now, we know that there's utility in terms of using this information to help decision making, but does it change anything or have any effect on improved outcomes when it comes to fertility? The jury is still out. But we're very interested in doing the studies to find that out.
Yeah, so it would be really interesting to see. And my gut reaction to it would be that a lot of these polygenic conditions that you listed that's included in this have adult onset or late adolescent onset. So it kind of makes sense that maybe, right away we wouldn't see a change. But I mean, we know so little about genetics when we really think about it. So it'd be interesting to see the results.
It's so true. And you bring up a really, really good point, because there's this whole other issue with something called, that I've been learning about, as this definitely not information that I had years ago, but the more I also, as a genetic counselor, delve into, how polygenic, diseases work. But there's this whole issue of pleiotropy, which is sort of, general good health, good health for one thing impacts good health for another thing. And some of this is sort of intuitive, for example, we're very well aware that if we have high cholesterol and high blood pressure, we also have a higher risk of, coronary artery disease. They go together. And those polygenic risk scores go together too. And so what we don't know, though, is, if we're looking at these polygenic risk scores, is there something to it that we don't know about that may have an earlier effect, and maybe even an earlier effect, all the way down to, implantation rates? That we do not know yet. But yeah, that's the curious piece, because yes, a lot of these conditions are adult onset conditions and are conditions that, impact later in life. But we definitely do know that these conditions impact each other. And there's sort of a, this, snowball effect, and that these variants don't necessarily, these risk scores don't operate independently. And that's part of the reason also we're trying to come up with this index, because looking at these scores independently have less utility than looking at the scores sort of as one global number. There's a little bit more utility, and the data is stronger when we look at it together versus separating out from into specific scores for specific diseases. It's very interesting. And the more I learn and the more we learn as a group at the lab, the more fascinating it gets.
Yeah, it certainly does. Anything genetic related. And I think with polygenic risk scores being so much in the news and new research happening in the last few years, I think that this is such an important piece of that, and seeing what other research comes out of this. And the more we learn, I do want to end with a question about what a lot of patients are probably thinking about at this point. It sounds great, but how much does it cost? Is insurance playing any role in this? It's such a new technology. What would be maybe an out-of-pocket cost for a patient that wants to pursue this type of PGT?
That's a great question. So for us, the costs of, in general, the costs of PGT in general can range from a few hundred to a couple $1,000s, depending on what a family is requesting. In our laboratory, we don't charge anything extra. For example, if a couple was coming to us for monogenic testing and then also wanted to do polygenic testing, it would actually be the same cost.
It'd be the same cost if someone's already doing IVF with PGT. Wow.
With PGT, yes, with PGT-M or PGT-SR. And so the costs are a little bit more if they were just doing aneuploidy screening, because it's more involved analysis. But if a couple was coming to us for PGT-M or PGT-SR and said, oh, I'm also interested in the polygenic testing, they would not actually be paying anything extra. So generally, though, for PGT-P studies, it would be a few $1,000s.
I definitely wasn't expecting that. I thought you were going to say much more to do this whole level of, you're looking at so much more data, as we keep saying, than if you're just looking at aneuploidy of counting the chromosomes. Okay, does this one have an extra 21 that would lead to Down syndrome? Or looking at that one gene, is there any variance in that gene that could be pathogenic? But here you're looking at so much. So it's really interesting to hear. And with so much fertility insurance coverage changing in the last year or so, and I think in New York, there was a new law at the beginning of January 2020 to be more inclusive with insurance coverage, that I imagine, and hopefully we're going to see more and more insurance coverage with, IVF with PGT, because that seems to be the most common route.
Yes, I hope so. I mean, yes, and you hit the nail on the head. Right now with insurance and PGT, it's a very not all insurances cover it, or they cover a certain amount, and not even, it's very complicated, and it's not always covered. And I would love to see, in the future, we're definitely not there yet, but in the future where we're able to, work directly with insurance companies and get, this type of testing, covered, because the overall goal is, and what insurance companies love is, well, does this save money? Is this money saving? And like, theoretically, yes, over time, if we have the opportunity to reduce, disease risk, then the overall disease burden, as a population, would not be as high, which would be a benefit, not just to insurance companies, but of course to, for us all. So I would love to see that. But right now, as it stands, if somebody were to come to our laboratory tomorrow, insurance is not covering polygenic testing. It's just too new.
And that's something we'll keep our eye on. And as we end here, I do want to say that it's important for patients to be pursuing genetic counseling along with their process of if they're going through PGT, to be educated on this. Podcasts like this are great to learn more background information, but it's definitely going to depend on a patient's personal situation, family history, all the things that we go over in genetic counseling.
Yeah, absolutely. That is 100%. And anybody coming to our laboratory for this type of testing, that's actually, we're very pro genetic counseling, and it's a requirement, actually. Everybody speaks to me at this time, and they all get a full genetic consultation regarding this testing, so that the benefits and limitations can be discussed in detail, because that's critical.
Yes, that is very vital to this process and informed consent to understand all of this, because it's quite complicated, as we've seen in this series so far and in this episode. So thank you so much for coming on and explaining this new technology. I definitely want to keep my eye on this and just see all the new research that comes out of this. And, this may end up being that PGT is becoming more and more standard within fertility treatments, and that maybe we'll just end up doing all types of PGT on each person in the next few years. Very interesting to keep track of. So thanks for sharing with me and listeners today. Really appreciate it.
Thank you so much.
Learn more about genomic prediction and their PGT-P on their website, genomicprediction.com. You can also follow them on social media by searching Genomic Prediction. For more information about this show, you can go to dnapodcast.com. We've got over 120 episodes on there that you can stream. And any questions you have, there's a contact form on the website, but you can also just directly email into info@dnapodcast.com. So any questions you have for Jennifer, myself, or any of the guests that's ever been on the show, send into info@dnapodcast.com. And you can connect with us on Instagram, YouTube, Facebook, Twitter, all the social media. Just search DNA Today Podcast. Please take a moment to rate and review this show on Apple, wherever you listen to this podcast, so that more people can find it. Let's Get Checked offers a variety of home testing kits, including fertility, but also sexual health with STI testing. According to the World Health Organization, of the 19 million Americans who contract a sexually transmitted disease each year, only 50% are aware that they're infected. Only 50%. People who are sexually active should all be doing STI testing on a regular basis. Luckily, Let's Get Checked makes it easy by sending a kit right to you in discreet packaging. It's just like the other kits I've talked about. You collect a sample, pop it back in the mail. Wait time is quick with results in only 2 to 5 days. As most of you know, Let's Get Checked also provides fertility testing too. If these episodes make you curious about your own fertility status, you can start learning with their kits. I did the female fertility test and had my sample run in their CLIA-certified lab to know more about my hormone levels for specifically follicle-stimulating hormone and luteinizing hormone, and a bunch of others. Try any of these kits for yourself by going to letsgetchecked.com and use code DNA Today for 20% off your kits. Again, you can use code DNA Today for 20% off your kits at letsgetchecked.com, and that code works for any of the sponsors from this show. DNA Today is the code to be getting all these exclusive deals as a DNA Today listener, and a thank you. Join us next time to learn and discover new advances in the world of genetics.
The genes of you and me, the genes of you and me are all made of DNA. We're all made of the same chemical DNA.